Okay. Happy Friday. Aloha. Good to see you. Hopefully we can do this in person next week. I think our numbers are actually down this week, so I think that's probably a good thing. And we'll just need to keep them down and not go crazy this weekend. So we have 2 papers to talk about, and we have an exam on Monday, and I wanted to do some review if anyone had questions. So what do you want to do first? Does anyone have like impending questions for review that we want to tackle and then talk about the papers, or you want to try and run through the papers and use whatever time we have left? Any thoughts on which way to go? Does anyone have review questions that they want to ask Allie? Can't hear you, Allie. It says you're unmuted, but I don't know. Anyone else have questions?
I have some questions. For me, let me see if I can find that. So we have this that you gave us. It's the feeding and satiety stuff. And I kind of made sense of it, but for me, it's just really helpful to actually see you draw it out, like as you're explaining it. Is there any way you can do that?
Yes. I guess. I don't know. No, I don't know the answer to that question. I mean, I don't have something I can draw on. Trying to think about the best way to do that.
You can draw on the screen. If you share screen and just share a white screen,
yeah.
Then when you share screen, there should be like a thing that says annotate, and you can draw on the screen.
I'm not gonna be able to do that with a mouse. I can tell you right now. Sorry. I hear you, Kate Reed. Can I just go over it? How about I just walk through it? Can we do that?
Okay, that would still be helpful.
Okay. And Allie, I sent a note. Put your question in the chat if you can hear me, and I'll answer it. You can type it out in there. Okay. Okay, can you all see that? All right. So we have, and can you see my mouse too? Moving around? Okay, so I'll use that in a minute. So the arcuate nucleus, right, essentially regulates the 3 areas of the hypothalamus that are involved with different types of feeding. So lateral hypothalamus basically does eat or don't eat. Ventromedial hypothalamus does numbers of meals per day, right? So more meals when you're more hungry, less meals when you're less hungry. And the PBN does the meal size, the size of the meal, right? The length of the meal. And so we have, so we have the NPY, AGRP neurons, and they are going to stimulate appetite over here, right? Stimulate appetite. At the same time, they're going to decrease metabolism, right? So they have this sort of biphasic effect. They're going to make you hungry and conserve your energy. The POMC cart do the opposite, right? They're going to decrease appetite and they're going to increase metabolism. So that's why you see, right, these inhibitory and excitatory things. Now, the thing is, so why are these excitatory from POMC? Well, that's because when you excite the paraventricular nucleus, you decrease meal size. And when you excite the VMH, you decrease the number of meals. When you inhibit it, you increase the number of meals. So it's kind of backwards. I didn't make the system. This is how it works, right? For eating, it's the opposite, right? So the POMC is going to shut down eating in the lateral hypothalamus, and NPY is going to stimulate eating in the hypothalamus. So are we good on the way in which the arcuate nucleus controls the other three? Good? We're good? Thumbs up? Yeah? Okay. All right. So what controls the arcuate nucleus? And that is going to be all these different peripheral signals, right, that are coming in to sort of regulate our hunger and satiety. And there are things like ghrelin and glucagon, which are going to signal hunger. So the longer we go without eating, the more ghrelin builds up. And so ghrelin's going to stimulate NPY and inhibit POMC. We have satiety signals like high insulin. Insulin's high after we eat when we have high blood sugar. PSY330 is high after we eat. That comes from the gut, right? And so those are going to stimulate POMC cart and shut down NPY. And then those are sort of our, we would call them our short-term signals, right? Because they're regulated by, did we just eat, or how long has it been since we ate, right? Then we have a long-term signal, which is our fat reservoir, right? That's our leptin levels, right? And so leptin, when leptin is high, that means we have higher amounts of fat. And so that is going to be a satiety signal. So that's going to stimulate POMC cart and decrease NPY, right? When leptin levels drop, that signals that we're using up our fat reserves. And so that's going to stimulate us to eat, right? So that's going to act on NPY to stimulate that and to decrease POMC cart. So I think that's everything. Does that help, Kate Reed?
Yeah, I think I followed everything except the like where you have the appetite and metabolism. So I guess just for like NPY, say it increases appetite. So is that where it's exciting the LH, and then it's inhibiting the VMH and the PVM because inhibiting them increases those things?
Yep.
So then where does the decreased metabolism come in?
Well, those are different pathways. So I'm just saying that it can also decrease metabolism, but this particular chart is just for appetite.
Okay.
Okay. Lena asked, do we need to know the research articles for the exam? You need to know, yes, you need to know the overall like point of them, right? So I'm not going to ask you like to tell me about the data they collected, or I'm not going to be nitpicky about it, but you need to understand like the take home message. And I'll try and when we go over today's articles, I'll try and emphasize like what kind of question I might ask from these two articles. Other questions?
What's the format of the exam?
It's all free response questions. And did you all see the exam review questions that I posted on Moodle? Right, so those are examples of the types of questions I might ask on the exam. So this class is really like, I want you to get concepts, but I want you to have details to support the concepts. So what we just talked about is a prime example, right? You need to understand the concept that the arcuate nucleus is controlled by peripheral signals, and it controls feeding, but then you need to know how it does that, right? Kate Reed, you were gonna ask a question?
Yeah, on the review questions, the very last question, it talks about hormones, the reproductive system in both male and female systems. And then it asks how stressors can contribute to infertility. I guess I was trying to narrow down that question because there are like a million ways that, like, do you want us to talk about ED or pregnancy, or do you want us to talk about like delayed ovulation and decreased sperm production, or is it just all of the above?
Yeah, I think that the last slide of that lecture, this one sort of summarizes that, right? For males and for females, that's sort of overall. But yeah, I mean, understanding, okay, what is the role of LH and FSH in females and in males? And then how does stress impact those hormones, and what is the behavioral outcome, right? So like 3-part question.
Can you repeat the 3-part? You said LH and FSH.
So what do they do normally in
Right.
women and men, right? And then how does stress affect those levels? And what is the behavioral outcome of stress affecting those levels? Allie.
Can you hear me now?
I can.
Okay, cool. So if we feel that we can answer a question with a chart, is that acceptable?
Yes. I do not need like complete sentences. If you want to draw something, if you want to put bulleted points, if you know, however you want to do it.
Okay, awesome. Thank you.
Any other questions, or you want to get on to the research?
I have one last question if nobody else does.
Go for it.
There was just one question on the review that I didn't know where to find the information for. I think it's the 5th question about perception of control and long-term health effects.
Yeah, I'm not sure I fully found the right answer for that as well.
Is that part of the stuff that's in the cancer section of the book that you said to just take it from the book?
Yeah, it is. I won't put that on the exam. So we didn't really cover that. I mean, this review is, I didn't write this review this year. I mean, this is from a previous year. So yeah, don't worry about that question. So the one on the perception of control, that won't be on there. We're going to talk about that towards the end of the class when we talk about how to manage stress and ways to reduce stress. Anything else? Okay. So I want to talk about the Graves' disease and toxic nodular goiter papers first, but I have lost my Zoom. There you are. Okay. And I'm going to share my screen. So let's start out with what is the what is the difference? Who wants to answer? What is the difference between Graves' disease and toxic nodular or TNG? Why are they looking at these two things? Yeah, Allie.
So TNG is the excess of thyroid at the tissue level because there's like too much hormones.
Mm-hmm.
And so they're looking at whether or not stress can basically speed up this process for both Graves' disease and TNC or TNG.
Yeah, so you're right. The TNG is a hormonal issue. And what causes the Graves' disease? We talked about that in class. Autoimmune, yeah. And so it's thought that why does the autoimmune get triggered? The thought is that stress can play a role in creating that, right? And so that's sort of their experimental manipulation. And the TNG is kind of their control, like should not be stress. We don't think stress causes that. We think that it's a hormonal imbalance, non-stress related, right? So that's kind of, and then they have a control group that doesn't have anything. But the reason they're including that other disease state in there is that they think it's a non-stress, but same system, right? Okay. All right, so what did they measure?
You want somebody else?
Yeah,
to-
Mina.
They looked at stressful life events within the past 12 months.
They did. All right, so I'm gonna share my screen. Maybe you guys over here. So they use this this scale, right, of life stress inventory. And so I'm gonna try and pull it up. Okay, so real quick, run through and write down the point scores. Hopefully nobody had death of a spouse in the last year. But if any of these things happened to you in the last year, been kind of a stressful last year. I'm not gonna lie. We might have a little elevated scores right here. But go through and just write all your points down and then get your calculator out and add it up. I already did it for me. I'll share my score. I scored 233. Okay. How are we doing? I'm going to launch a poll and it's anonymous, right? So when you're done doing your points, answer what your score was, what range it was in, and we'll see where we are. And that way I'll know you're done. So that's going to be my signal that you have added all your points up when I see that everybody's participated in there. All right, need 3 more. Oh, 1 more. All right. Everybody's done. Good deal. All right. So look, those results are kind of interesting. We got a little bell curve there, right? We got 2 people on the low side, most everybody's in the middle, and 2 people on the high side, right? So as you can see on okay, done with that. All right, now all these windows and stuff, I still can't manage Zoom. All right, so in general, right, this is going to say, this scale says that if you're under 150, you have a low amount of life change. And you notice some of those are like positive changes, like vacation. But going on vacation can be stressful, right? Or outstanding personal achievement. You think that would be good, but often those are associated with levels of stress, right? And So it looks like most of us need to be really practicing healthy lifestyles because we have a 50% chance of some sort of major health event in the next 2 years, which is probably not surprising given how much stress we've been under for a year now. So anyway, that's what they did. All right, back to our paper. That was our fun for today. All right, who wants to take a stab at table 1? Explaining table 1.
I can do it.
Lena, okay.
So table 1, so the life experiences survey looked at the impact of events participants had experienced and whether they view those events as positive or negative. So we can see that for Graves' disease, there's more positive and negative events than the non-autoimmune disease, TNG. And also more than controls, but the significant part is that there are more negative events with Graves' disease.
Yeah, right there where my arrow is. So, and it's kind of interesting that Graves' disease overall had like 100 more events total. So like these people are not only, they have more positive events, but those can be stressful as well, right? All right, who wants to tackle table 3? Oh. Yes, Evelyn.
So in table 3, it talks about the impact of the 2 types of events, the positive and the negative one. And it's talking about the perceived impact. So people with Graves' disease, they saw or they perceived the impact of negative events as higher than the TNG and the controls, which the numbers like almost double compared to TNG. But for the positive, it wasn't as high, but it was still higher than the TNG and controls.
Yeah. So not only did the Graves' disease have higher numbers of events, but the impact of those events was also magnified. So it's almost like a multiplier, right? All right, table 5. Who wants to tackle that? What's the point of table 5?
I can take that one.
All right, Philip.
So table 5, they're looking at, so they said within the past year, but they're breaking it down even more to within the past 6 months versus 7 to 12 months ago. And what they found was that a lot more, especially for Graves' disease, a lot more of the events that they remembered were 7 to 12 months ago. And that's prior to the symptom onset for a lot of them. And so that supports that these negative and positive stressful events might be a cause rather than a consequence of the disease. So it's not that the disease is causing the stressfulness, but maybe the stressfulness is causing a flare up in the disease.
Absolutely. All right. Any other questions on this one? We're good? All right. So I'm trying to think up. I know this isn't on this exam. This is on the next exam. But if I was going to ask a exam question about this paper, I might ask, what is the relationship between stress and Graves' disease and toxic nodular goiter disease? And I would want you to tell me about the number of events, the differences in the number of events, and the impact of those events, and when they happened. Right? Those are sort of the 3 big things, takeaways from this paper. Right? And you wouldn't have to know the numbers, like just saying there were overall more positive and negative events, that sort of thing. But I wouldn't expect you to say there were 355 negative impact, right? So not that level of detail. Okay, next paper. Social ties and susceptibility to the common cold. So what's the premise of this study?
they wanted to know if having like a greater variability of social ties decrease the risk of you catching the common cold.
Yes, exactly. Let me get to it. And so they said that there's a this hypothesis that, you know, greater connectivity leads to better health outcomes, and in particular that they said that that it had been reported the relative risk for mortality among those with less diverse networks was comparable to the same relationship between smoking and mortality, right? So they're really trying to make this like it's a big deal, right? So what did they, what was the design of the study? What did they measure?
There were 12 social tie groups.
Yep.
Yeah, and so they also looked at, like, for example, people who smoke, people with poor sleep, people with decreased like vitamin C intake, introverts, and looking at like the social aspects of things. And then they like nasally in the nasal passage or whatever sprayed the common cold virus and saw who was more susceptible to getting sick.
Yeah. So they had like sort of two categories, right? The social network diversity, which they looked at these 12 different groups and people's engagement within these 12 different groups. And so this is the actual scale, right? And we're not going to fill this out because it's a little bit more detailed. And there's actually a scoring index, like you have to score like on, I think for how many children you have to score like at least 2 or something. I don't know. They have like a whole scale of like what qualifies as being in a network, right? So they fill out this social network index, right? And they end up with these scores, and you're either low with a score of 1 to 3. The maximum score is 12. And so they're low, moderate, or greater than 6. They scored as high, right? Then they also looked at a bunch of health outcomes that might be related to your risk for a cold, right? Like exercise, smoking, vitamin C, the things that Allie mentioned, right? They also looked at pre-challenge antibody. So what does that mean? Why were they doing that?
They were looking to see just the antibodies, the individual differences in antibodies that people had already built up prior to their exposure to the rhinovirus.
Yeah. So they sort of grouped everybody in the study as you either don't have a lot of antibody, that less than 2, or you have more antibodies, right? So what was the sort of point of that? Well, why would that be important? Did you say that?
These are associated with your like immune system.
Yeah. So what would you expect for the low versus the high and contracting the cold?
Less antibodies means you're more susceptible to getting sick.
Right. So they wanted to sort of separate out people and see if that mattered, right? Okay. Then they gave everybody the infection, right, and measured the percent. And so here we see in red, right, those 2 red boxes, those are percentages of the total. And we'll just look at the totals. We won't get concerned about the Hanks versus the RV39. But, and so that did matter, right? If you had greater than 4 antibody score, only 70% of those people got infected, but 99% of the people under 2 got infected, right? So you didn't, they defined cold, right? Not just as whether or not you got infected, but they measured the behavioral symptoms, right? So they had this objective criterion, which was like them measuring things. But then they also asked people, do you feel like you got a cold, like you contracted a cold? So they had both an objective and a self-reported criterion. And we're just going to sort of ignore the Jackson criterion, right? So again, looking at the percentages, even though 70% of the people with the greater antibody tier got infected, look at the percentages of them that showed signs, right? So not only does having a higher antibody load protect somewhat from getting infected, but it really protects you from experiencing that infection, right? Which is what I talked about in our lecture with the coronavirus and the vaccine, right? Is that the vaccine doesn't keep you from getting infected, right? The vaccine keeps you from getting sick from getting infected, right? So you get infected and your body can quickly get rid of it before you show any symptoms or signs of that, right? But that's also why for a while, until we can get rid of the coronavirus, we're still gonna have to wear masks, because even though you've been vaccinated, if you are carrying it, you can be putting it back out in the air, and other people who aren't vaccinated then, can contract it. So I think that's important. I don't think people understand that. They think once they get vaccinated, they're like immune and it's all good. All right. Where am I? We did table 1. All right. So what were the results of social network and susceptibility?
People with lower social network diversity had a higher percentage of colds, and then people with the higher network diversity had a lower percentage of colds. And it was this, we saw the same trends for the objective and the self-report.
Yeah. And so the key point here is that it's the diversity of your network, not the number of members in the network, right? So your contact with numbers of people, like your number of relationships, did not correlate with this, did not show this effect. But the diversity of those relationships is what was important to show this effect, right? What about what health practices put people at risk of a cold?
You mean like
Dr. Pittman,
Yeah.
I had a quick question.
Uh-huh.
Go before.
Go ahead, Ash.
Go ahead.
So with the results, when it said the highest rate of colds was associated with those with high school degrees or less, and then it was some college, or the next was with college degrees, and then the lowest was with some college, could that be related to stress of like higher jobs? Because like
Sure.
the stress of like with high school degrees, you have like not lower jobs. You understand what I'm saying?
Yeah, no, I do. We haven't talked a whole lot. We're going to talk in this next exam section about socioeconomic status and the relationships of socioeconomic status and stress and health outcomes. But certainly, I think that's what they're looking at, right? So people with high school degrees and no college degree are probably in a lower socioeconomic status than people with college degrees. Yeah, I think that's the thing that's probably, and higher stress is typically associated with lower socioeconomic incomes than higher.
And I had another question about like the numbers of like diversity. I know in another class we talked about like it's better to have a few really close friends than have a lot of acquaintances. would like the bond of the relationship have a bigger effect than just have like a few acquaintances that you talk to, but like talk to on a rare basis?
Yeah, so they didn't look at the quality, right? And that would have been interesting to add in like quality as well. Like, so I'm sure that there were, I mean, you can only look at so many things in one study, right? But you could imagine that there is, I mean, you look at the standard error bars for the low group there on the screen, and there's more variability in the low group than the moderate or the high. So wouldn't it be an interesting question to look within the low group and say, okay, those of you that were more likely to get a cold versus a group that's less likely, what is the quality of those relationships that you have, even though there are a few number? I would predict, right, those that were down here, right, causing the variability to be down here, that group, even though they don't have much social network diversity, they probably have high quality relationships with those individuals. That would be my prediction, right? So yeah, but they didn't look at that. They were just looking at the diversity.
And they qualified like if you had a relationship in a different social network, you had to talk to them at least once every 2 weeks. So I mean that's not that strong of a relationship.
Yeah, but they did that scale that I showed you, you know, like I said, it has a scoring on it. So there is some measure of like, it can't just be acquaintances. Yeah, I know people in all 12 groups, therefore I'm a 12, right? It has to be an engagement in those groups at some level, right? Okay. So back to my question, which is what health practices put you at risk? So remember, they did the social network diversity, but they also asked about all these other things. Allie, you want to do that one?
Yeah, it's like the things that I mentioned before, people who smoke, people who have bad diets, people who have poor sleep patterns, lower vitamin C levels, alcohol abstinent individuals.
Yeah, that was sort of interesting. Would you predict that the people that drank less would be more likely to get a cold?
I think in my mind, I was thinking more like people who used to have alcohol problems and who are now abstinent from alcohol. It could put a lot of stress on their system because not only are they going through withdrawals, but like they don't have that social escape anymore, I guess.
I was sort of thinking of it as like, I mean, hopefully most people don't drink alone, right? So if you're consuming more than, I mean, nowadays, but anyway, pre-COVID, when we all would socialize, that was sort of a social thing, right? And so maybe that was like a confound, right? That people who had more than one drink a day also were in the higher social network diversity group, right? So it wasn't necessarily the alcohol per se, it was the social network aspect kind of bleeding through that, right? Sort of my thought on why that might happen. What did they find out about levels of norepinephrine? That was interesting. Why do you think they measured that, and what did they find?
Didn't they find that the higher levels of epinephrine, like above the median, were at a greater risk for the colds than those below it?
Yeah, they did. So why do you think they would have measured that, or what do you think the significance of that is?
It's like one of the, oh, it's not like one of the stress hormones, but it's one of the catecholamines that are activated with like the sympathetic nervous system.
Yeah, yeah. So higher, sympathetic nervous system activity probably means more stress, more anxiety, and so those people were, that's probably getting at the link between stress and immunosuppression, right? And the likelihood of getting a cold. Right? All right. what about this figure? So then they took and sort of grouped the low and moderate people together and separated out the high group. So they put the 1 through 5 together and the over 6 together, right? And they looked at whether or not that, pre-challenge antibody level made a difference. And what did they find there?
Social network diversity is a protective factor in all people, regardless of the amount of antibodies that you had pre-exposure.
Yeah. So again, higher number of colds in the low exposure group than the high exposure group, but having the higher networks, right, was slightly lower for both of them. I mean, it's not like a huge percentage. Sorry, it's not a huge percentage, but it is slightly lower, right? All right, and let me stop my share. Last thing, what did they find out about personality? What did they talk about with personality?
Someone sent a question, by the way.
Oh, sorry, Joe. Let me see what you got. So Joe says, going off this study, do you think the lack of social interaction diversity during COVID is spreading the virus quicker or making people more susceptible to it? What do y'all think? I think that probably staying more isolated is protective from catching and spreading the virus at this point than the detriment of decreasing your social network diversity. But certainly, yeah, I think that could maybe come into play. I think all the things that we're doing right now, like using hand sanitizer and washing our hands and wearing masks and wiping down surfaces before we touch them. I haven't had a cold this year. I don't know if y'all felt healthier, but I think this transmission of a lot of airborne sort of things is way down because we're doing all this stuff. So yeah, I think all the physical stuff we're doing probably is keeping us healthier than the fact that we're not interacting maybe with as many people. All right, last question, personality. Anyone want to take that? They didn't really talk about it till the very end. They didn't even really mention that they were measuring it.
They looked at extroversion of the big 5 was the only one that they found an effect for, and people below the median of 28.5 of introverts were more susceptible to colds.
Yeah. And they found that being extroverted correlated with having a more diverse social network, right? Which I guess makes sense. I mean, is that a confound? I don't know. It's hard to tease those apart, right? I mean, you would need to have a group of introverts that had high social network diversity and a group that had low social network diversity and look at that. And then a bunch of extroverts that were in small groups versus diverse groups and sort of look at that in order to try and tease it apart. But I don't think they had like enough data to really do that sort of stuff. So, all right. If you don't have any other questions, that's all I got on these studies for you. Or if you have a question about exam stuff, we can go back to that. Yes, Lena.
I just have like a general question. So this study was conducted in 1940 something. And the participants that mentioned that they received $800. Would that still be possible today? Injecting them with virus?
Oh yeah.
Yeah.
No, there's like a whole Institute of Cold Research in England, and that's all they do is like give people colds and look at various things. Now, I don't know what they pay them. I mean, that sounds like a lot of money for 1940, but I mean, if you're going to get infected with something, I think you gotta get paid pretty well.
Did you guys see that? It was like a footnote in the ulcers book or whatever, and it was like talking about this study done in Germany on colds, and you could basically get like your flight paid for, like a 2-week vacation, and they would just like inject you with cold virus, and you might get it, you might not, but it was basically like a free vacation.
Yeah, I mean, on a side note, I think I told y'all that I participated in the Moderna trial, the vaccine trial, and they unblinded it last week, and I was in the placebo group. So they gave everybody in the placebo group the vaccine, which was nice. So we ended up, everybody in the trial ends up getting the vaccine eventually when they unblind it. But just on a side note of that, I'm still now that I got the vaccine, I'm in the vaccinated group, and they're still studying us and our rates of infection, stuff like that. So I'm still participating in the trial. But since I guess I started at the end of August through January, they've probably paid me about $2,000, and I got the vaccine. So, I mean, that's pretty good. Sometimes being in these trials is a pretty good thing. So, all right. Nothing else? Yeah, and this is an old study, and I chose it because of the relevance to what Sapolsky. I mean, I'm trying to find some studies that he actually references in the book so that we can see the primary literature on that. And it was pretty easy. I gave y'all some hard papers last time. All right. Y'all don't have anything else. We're good to go. And I hope and pray I see you on Monday at 10:30. And we probably won't find out until 4:59 again today. But look for that email. And if it says we're going to be remote, look for, I'll freak out and probably need 24 hours to calm down. And then I'll send you an email about what we're going to do next week. All right. Stay safe and well.
Thank you.